Hello everyone. Today's topic is chemiosmotic hypothesis. What we are going to learn in chemiosmotic hypothesis. As you know, photosynthesis has got light reaction and dark reaction. During light reaction, there will be preparation of ATP and NADPH. During dark reaction, ATP and NADPH is utilized to fix carbon dioxide. This is the basic concept of photosynthesis. How ATP is produced? Where it is produced? In the chloroplast. How ATP is produced in the chloroplast, that is explained through one hypothesis known as chemiosmotic hypothesis. It was proposed by Peter Michel. He says that ATP is synthesized in the chloroplast because of the proton gradient force that is developed. What is meant by proton gradient? Gradient refers to when one side concentration is more, another side concentration is less. Proton gradient refers to when one side H plus concentration is more, another side H plus concentration is less across the membrane. Then that is known as proton gradient. This proton gradient develops a force. That force is responsible for synthesis of ATP. That is the whole concept of chemiosmotic hypothesis. We are going to understand in detail how exactly ATP is synthesized using proton gradient force. Before that, let us understand how is the structure of chloroplast. To explain structure of chloroplast, as you can see, I have drawn a double membraned chloroplast in green color. This is the membrane of chloroplast. Inside chloroplast there is stroma, and instead of showing all the thylakoids and grana, I have shown a single thylakoid with the membrane. This entire thing is a membrane, lipid bilayer. It is shown in pink color. What are the things associated with the thylakoid membrane? Thylakoid membrane has got photosystem 2, whose reaction center is P680. It is nothing but a specialized chlorophyll A molecule, and an electron acceptor known as pheophytin. Pheophytin is an, here I have written, colorless pigment. It is electron acceptor. Then there is plastoquinone, PQ I have written. Plastoquinone is a mobile carrier, mobile electron carrier. What is mobile electron carrier? It can move freely in the membrane. It is not fixed at one place. Then there is one more complex, cytochrome B6 and cytochrome F complex. Then there is one more electron, mobile electron carrier known as plastocyanin. What is the difference between plastoquinone and plastocyanin? Difference is plastoquinone contains ferrous, hence it is red in color. Plastocyanin contains copper, hence it is blue in color. Apart from that, these are the two mobile electron carriers located in the thylakoid membrane. Important to notice. Uh, then there is photosystem 1, whose reaction center is P700, and an electron acceptor FES. There is FD, ferredoxin. Ferredoxin is not a mobile electron carrier, remember. It is just attached to the photosystem 1. Then in the membrane of thylakoid, you will find one enzyme, that is NADP reductase. In thick pink color I have shown. Apart from that, what else is there? In the membrane you will find F0 F1 particle. F0 F1 particle, it has got a stalk and a head. The head of F0 F1 particle is, it behaves like enzyme ATP synthase. How, if F1 F0 F1 particle is also found in mitochondrial inner membrane, how to differentiate? While writing chloroplast F0 F1 particle, you can use C F0 C F1. C refers to chloroplast. That's how you can differentiate. This is about the thylakoid membrane. Then within inner to the thylakoid membrane, there is a space which is known as lumen. Associated with photosystem 2, on the inner side of the thylakoid membrane towards the lumen, there is water splitting complex. I have written it as WSC, water splitting complex. These are the details of diagram. Now let us understand, during chemiosmotic hypothesis, what are the three things that is responsible for developing proton gradient across the stroma and lumen. You have to compare stroma with that of lumen. How lumen builds up more H plus, how stroma H plus decreases, that's what we are going to learn. To understand that, let us know the non-cyclic photophosphorylation Z scheme. During photophosphorylation, light is absorbed, light will be having photon, therefore one photon is required to stimulate P680. When photon stimulates the reaction center of PS2, there will be ejection of electron. One electron is ejected, it is accepted by electron acceptor pheophytin. Hence, now P680 is deficient of electron. That will keep it in mind. Pheophytin transfers its electron to mobile electron carrier, that is plastoquinone. Plastoquinone, after accepting electron, now it will transfer its electron to cytochrome B6 and F complex. It can, because of the energy of the electron, now plastoquinone, it will take up proton from the stroma. Remember, it is from the stroma, H plus ion it is taking. But H plus ion cannot be transferred further. Why? Because there is electron acceptor, but proton acceptor is not located in the thylakoid membrane. Now what to do with the H plus ion which is being taken from the stroma because of the electron energy? Whatever H plus ion is taken, that is actively pumped from the stroma to the lumen of the thylakoid membrane. So what plastoquinone is doing? It is taking up the H plus ion from the stroma, giving it to the lumen, pumping it to the lumen. Hence what is happening? The H plus ion concentration of stroma is decreasing, lumen is increasing. That is the first reason for developing concentration gradient. I have written it as plastoquinone. What it will do? It will take up the H plus ion from the stroma, gives it to the lumen, hence reduces H plus ion in the stroma, responsible for developing gradient. From cytochrome B6 and F complex, electron now comes to the, it is accepted by plastocyanin. Plastocyanin gives its electron to the reaction center of PS1, that is P700. P700 will also be accepting one photon. Therefore, for the transfer of one electron, how many photons are involved? One to stimulate reaction center of PS2, one to stimulate reaction center of PS1. Two photons are involved in transferring one electron. I have written one electron dash photons. It is two photons are involved. That we will keep it in mind. Now electron has reached P700. P700 ejects its electron to the FES. FES is an electron acceptor of PS1. FES transfers its electron to ferredoxin. Ferredoxin will transfer its electron to the thylakoid membrane where there is enzyme located, NADP reductase. What does NADP reductase do? NADP reductase, it will pick up one NADP from the stroma. To that, two electrons are added and a single H plus ions are added. What will happen to NADP plus? NADP reductase, now it will reduce the NADP plus to NADPH because it has accepted one H plus ion. This is how NADPH is synthesized. Hence, what is happening? For conversion of NADP to NADPH, one proton is taken. That is the second reason why stroma H plus is decreasing, because of the NADPH formation reduces H plus ion in the stroma. That is the second reason to develop proton gradient. Now what happens next? As I mentioned, P680 has ejected its electron. Now it is electron deficient. To fulfill that electron, water splitting complex, which is located towards the inner side of the thylakoid membrane, it will split the water into two electron, two proton and an oxygen. 2 electron, out of 2 electron, 1 electron, 1 electron is supplied to the P680 reaction center. It is not 2 together, 1 at a time electron is given. During splitting up of water, H plus ion again accumulates in the lumen. That is the third reason for building up of H plus ion in the lumen. That is, splitting up of water increases H plus ion in the lumen. Now here one question can be formed. We know that to transfer one electron, two photons are required to stimulate P680, one to stimulate P700. To split two water molecule, I have written here, for splitting two water molecule, how many photons are required? When two water molecule is splitted, there will be formation of 4 electron, 4 proton and 2 oxygen molecules. Now here we'll focus on 4 electron. To transfer 1 electron, 2 photons, that is to stimulate 2 electron, 4 photons. 2, if it is 4, then for 4 electron it is 8 photon that is required. Hence, to split 2 water molecule, amount of photon required is 8 photons. That kind of question can be asked, very important. Because of these three reasons. One is plastoquinone pumping, NADPH formation, which reduces H plus ion in the stroma, gradient is developed. Splitting up of water in the lumen builds up H plus ion in the lumen, gradient is formed. Because of these three reasons, there is building up of H plus ion in the lumen, pH decreases. That now H plus ion which is accumulated in the lumen try to go out back to the stroma. It can go back to the stroma only through F0 F1 complex. When H plus ion is moving, the force is generated, that force which it will convert ADP to ATP, which means ADP will combine with inorganic phosphate to form ATP. That's how proton motive force H plus ion which is going out through the F0 F1 complex. F1 is ATP synthase enzyme. It will generate ATP. So this is how chemiosmotic hypothesis explains how proton gradient develops, three reasons for proton gradient, and that proton gradient developed, how it ends up preparing ATP. That's about chemiosmotic hypothesis. Thank you. If you have liked it, please click the like button.